Hello, and a very warm welcome to everyone joining us for today's Select Science webinar entitled Accelerating Rapid Diagnostic Development with Optimer Reagents. My name is Cameron Smith and I'll be moderating today's presentation, and I'm also delighted to be joined by our guest speakers, Dr. David Bunker, Chief Technical Officer of Aptamer Group, and Dr. Edgar Goluk, Chief Executive Officer of QS Diagnostics. During this webinar, we'll be covering what an optomer is, the benefits of working with custom optomers in the discovery and development pipeline, working with optomers and their use in the development of rapid diagnostics, and how this technology can be leveraged to accelerate diagnostic and therapeutic development. Please feel free to submit any questions you may have during the webinar. To do this, simply hover over the speech bubble icon to the left of your screen and click to open the tool. So without further delay, I'm delighted to hand over to Dr. Bunker and Dr. Goluk, and would like to thank them both for presenting to us today.
Okay, thank you very much for the introduction. So my name's David Bunker, I'm Chief Technical Officer, and let's get started. So we've already covered the introduction for the presentation today, so I'm going to tell you a little more about what an aptamer is, what an optomer is, and then sort of things that you can do with them in a diagnostic context. So first off, some of the background to why people would want to use aptamers. So there are commonly known issues around antibody isolation and production downstream that can lead to various failures in diagnostic and therapeutic programs, even at very late stages in that process. So you want to try and avoid any of those issues wherever possible. There are also commonly known issues around the production and isolation that can lead to issues with security of supply. They can also lead to incorrect binding due to cross-reactivity or poor specificity, and it may not even be possible to generate an antibody against your target of interest. So you might go through the antibody cycle, as it were, time and time again and get nowhere. So these are the reasons that customers often come to us looking for an alternative. And that alternative, in our eyes, is an aptamer. So aptamers can actually be used as either a replacement or in fact as a complementary molecule to support antibody work. So for example, if your platform requires a sandwich pair, you may have a single antibody that you wish to use, but you can't get a secondary. The aptamer can fill that gap. Aptamers also have the advantage that they can be modified to make them useful for a whole range of different platforms. So this can involve putting functional groups to allow them to be immobilized on biosensors, for example. And we'll hear a bit more about that later on. As you can see on this slide, one of the biggest advantages of aptamers when it comes to biosensor development is their size difference. So you can see here, for example, that the aptamer is approximately 1/10 the size of the antibody in blue and purple respectively. So when you're looking at biosensor development, everything is going on closer to the surface, which generally leads to a more sensitive assay. We'll come back to that later. So just to fill you in on some of the basics of what an aptamer actually is. So an aptamer is a single-stranded DNA or RNA molecule that's been isolated to bind to a target of interest. Now in the body, DNA usually exists as a double-stranded molecule, essentially zippered together through base pairing. Now if you peel away one side of that molecule to leave a single-stranded DNA, there's the potential for that molecule to zipper up with itself and start to form some interesting secondary and tertiary structures, as you can see on the slide here. Now, not all of these molecules are going to be entirely double-stranded. That leaves single-stranded loop and bulge regions where those hydrogen bond donors and acceptors are exposed and available to interact with a target. For example, a protein, a small molecule, a cell surface receptor, etc. So essentially, an aptamer is a DNA molecule that's structured, and that structure allows it to interact with a target. And you can see an example of that on the right-hand side of the slide, a protein in purple interacting with an aptamer in blue. So how do we isolate an aptamer? Well, an aptamer library is made up of around 10 to the 14 different molecules, so different sequences that all fold into different shapes, different structures, and have exposed loops that can interact with the target. So we start with a library and we take a customer target represented in this case by the little jigsaw pieces at the middle of this diagram. We allow the aptamer library and the target of interest to interact under customer-defined conditions. So we try and keep these as close as possible to the end assay conditions. So if that requires a complex matrix, like let's say plasma, we include that in this complex formation step. We then partition aptamers that have bound to the target from aptamers that have not. So any non-binding sequences are removed. We wash the target to get rid of any weak binders and then recover the aptamers that are bound to that target. As these are DNA molecules or RNA molecules, they can then be amplified by PCR or reverse transcription and PCR to give an enriched population of binders. So this then forms our second generation to go round this cycle again. Now we're able to do additional things in this process, like build in what we call a negative selection step. So this is a step that's required to pull out aptamers which may bind to targets that are not of interest. So this eliminates the possibility of generating aptamers that cross-react with unwanted targets. So we can pull those binders out and dispose of them to give a specific population of aptamers. We then isolate individual aptamers from that and carry out a functional screen to find aptamers that bind to the target of interest. So what's an optomer? Well, an optomer is essentially the end point in our process of development. So what we have is, after we've isolated an individual monoclonal aptamer, we have something represented essentially by the middle diagram here. So we have a full-length molecule. Not all of that molecule is going to be required for target binding and target engagement. So what we can do is trim away those unrequired sequences to give a minimal functional fragment, which is what you can see on the right-hand side. And you can see by the scales underneath there that the aptamer can be in the region of 29-30 kilodaltons. The optomer can be as little as 5 to 10 to up to about 18 kilodaltons. Now there's a number of benefits to doing this. The first being cost-effective downstream production. So it's obviously more cost-effective to produce a smaller molecule. The other advantage of generating a smaller molecule, in biosensors, for example, this gets the interaction again a little closer to the surface, so it gives you a better signal, but it also gives better site accessibility on cell surface proteins, for example, better tissue penetration if you're looking at therapeutics and that sort of use. So this slide is just here to present some of the different sorts of targets that we can generate aptamers against. So you have the full spectrum of life here, everything from the very smallest molecules all the way up through proteins and viruses to bacteria and whole cells. We can generate aptamers against all of these different sorts of molecule. And at Aptamer Group, we have different processes tailored to fit the size requirements of the target. So our small molecule process is different from proteins, is different from cells. The table at the bottom I won't go through in detail, but this is just to give you some examples of the sort of molecule that might class as a small molecule, so an antibiotic or a drug, for example. A protein, that could be a viral protein, for example, or a cell, that's kind of obvious. And then underneath it, there's some of the applications that you might have. So, for example, biosensor development, ELISA development for proteins, and confocal microscopy or flow cytometry for cell targets. So that gives you a little flavor of what an aptamer is and how we isolate them. And I'll give you a few examples now of the sort of applications for aptamers in each of those different target areas. So the first one is using aptamers in flow cytometry and immunohistochemistry. So as I mentioned before, we can generate aptamers that are very specific to their targets. And we could do this with cells as well. So you can see in the flow cytometry data at the bottom left, we have isolated an aptamer here that very strongly recognizes the positive cell line on the left, but doesn't recognize the negative cell line, the control cell line on the right-hand side. So we have an aptamer that is specific to a live cell sample. What you can see on the right-hand side is the use of similar aptamers in imaging applications. So in this case, we've labeled the aptamer with a red fluorophore, Cy5, and what you can see quite clearly from this is these particular aptamers are targeting the protein of interest, but they're doing that in the context of a whole cell, and they have made it into the nucleus and are binding to the target there. So this is a good use of aptamers as imaging reagents and flow cytometry reagents. You can also use these, as I mentioned, in IHC. And in this application, they're almost identical to the way you would use an antibody. So the aptamer binds to a target on the surface of your cell or your tissue. The aptamer is then reported either with a fluorophore, or you can use the same system that you would use with antibodies. For example, a biotin on the aptamer capturing, for example, strep HRP and turning over a colored substrate. So you can use aptamers in much the same way that you would use antibodies. So that's talking about cells. Now we're looking at protein targets, and a very topical one. So in this case, we generated a pair of aptamers against different regions of the SARS-CoV-2 coronavirus spike protein. So we generated one aptamer that recognizes the S1 domain and a separate aptamer that recognizes the S2 domain. Now, using these two different aptamers, we can do exactly the same as you would do with a pair of antibodies. You can create a sandwich ELISA assay, and you can see an example of the concentration-dependent binding on the right-hand side that comes from this sandwich assay. The important thing here is to note that, one, you could use an antibody as one of those reagents, so you could use an antibody capture and an aptamer or an optimer to detect. Or you could use the pair of optomers as we've done here. Also importantly is the speed with which we can do this. So this pair of aptamers were isolated in just 17 days. So this can be a very rapid turnaround process. Small molecules are a little bit different. So small molecules, as the name implies, are very small. So generating an aptamer against these requires a slightly different process. So here what we do is rely on the flexibility that comes with the DNA backbone to allow the aptamers to wrap around their target. So they undergo quite significant conformational changes when they bind to their target. Now we can use this in a variety of ways. One way is to use it in aptamer selection, and another way is to use that actually in an assay format. So what you can see on the slide here is that we immobilize the aptamers, represented by the little red squiggles, onto a sensor surface. When we wash that surface, we remove any aptamers that are not actually associated with that surface, so that's background washing. And then when we introduce the target, the aptamers undergo a conformational change and they can be released from that sensor surface. Now these aptamers can then be quantified to give you a gain of signal. So this is a really great application of these aptamers because they give gain of signal responses from a single optimer binder. So we don't need sandwich pairs for small molecule assays. And just to give you an example of some of the work that we've done, so here we've got the example of an aptamer that we generated against irinotecan, a chemotherapeutic agent, and this aptamer does not recognize one of the metabolites, SN38. So you can see on this data set the aptamer very clearly binding to irinotecan, but not binding to the metabolite. So you have there a biosensor tool that can discriminate between one molecule type and a metabolite. So that's very important when it comes to drug regime monitoring, for example. And importantly, what you can see on the right-hand side there is that this interaction is concentration dependent. So you can use this to quantify the amount of a target molecule. And in this case, you can see that that concentration range spans the therapeutic window, the blue region highlighted there for this particular drug. So that brings me nicely onto the second part of the presentation, and I'll hand that over to Ed at QSM Diagnostics. He's going to tell you about some of the work that they've been doing with some of the aptamers that we selected. Ed, over to you.
Great, thank you. I'm a founder and CEO of QSM Diagnostics. As you'll see, our primary application for our diagnostic platform is to improve how infections are diagnosed. So the first part of my presentation, I'll highlight how we're developing this technology as well as how we're working with Aptamer Group to further expand our platform. And then I'll also describe some of the results that we've had with an aptamer or optomer targets that we've received from Aptamer Group. So my background is in chemical engineering as a person working in bioanalytical sensors. So some of the results I'll share today are also from my academic lab work that we have done to prove out this new platform. We have at QSM a strong group of individuals that have expertise in engineering as well as microbiology and biochemistry. And our first application is actually geared toward companion animals, so cats and dogs, looking at ear and urinary tract infections. But the platform that I'll be talking to you about can be utilized for a number of different applications. And hopefully by the end of this presentation, you'll be able to see how we could easily switch out the Optimer that we're using and be able to have a low-cost, simple platform that would give you an electronic readout in minutes using our collaboration with Aptamer Group. So the problem that we're addressing initially is for infectious diseases, for bacterial infections in particular. And this problem is true for humans as well as animals, but on the animal side, particularly challenging because you don't get feedback from the animals, and you need to do identification and be able to treat and diagnose based on the evidence that you can collect. So right now, the standard, what happens is, let's say for dog ear infection, you have a dog that's shaking their head, scratching at their ears. You bring them into the veterinarian. They'll look visually at the site and say, well, this looks infected or inflamed, and they'll prescribe a treatment that often includes an antimicrobial or antibiotic. And the problem is you don't actually know what's causing the infection. It could be yeast, it could be mites, it could be an allergic reaction, what's causing those symptoms. So at least in several cases, the veterinarian is able to collect a swab sample and put that under a microscope, stain it, and try to see what's happening on the microscopic scale. And they try to see if there's bacteria present and identify if they're circular or rod-shaped. But as you can see in the image, it's very difficult to distinguish. So the bacteria are so small, it's hard to confirm whether the bacteria are really there or if there are other particles that look similar. You can't tell if the bacteria are alive or dead. So this gives you some information, but it's not very reliable. And then the gold standard, what should be done each time, is to send out that swab to a laboratory where they would grow bacterial cultures, the same way they've done for 150 years. That takes several days, costs the pet owner over $100 to get those results. So in practice, that culturing test is almost never done, and you don't really know if you have a bacteria or what bacteria is causing the infection. And so practice goes through a trial and error process. So that's where our technology comes in. We have what we're trademarked as the Otter platform. It's a way to be able to detect and diagnose the cause of the infection, the actual bacterial species, while standing next to the animal. So we can go from a swab to swabbing that material onto our cartridge to results in 2 minutes, and to give that initial set of information as to what's causing bacterial, if there's a bacterial cause. We can diagnose if there's a bacterial cause, and it's also quantitative. So we're actually measuring biomarkers in that level and reporting that out. And that's important because you can use that value for follow-up visits to be able to say that the treatment's working or not, because the animal can't tell you. What we can't do currently is tell you which specific antibiotic to use, but that's okay. Resistance doesn't come up that frequently. You can use what's known as an antibiogram that we supply based on the species to with very high confidence, prescribe a targeted antibiotic that will work. And these antibiograms are updated regularly by regulatory agencies and at various clinics. And you can get the results this way and treat confidence. So how are we able to do this so quickly? So what we're using are what's known as quorum sensing molecules, or QSMs. And these molecules are secreted by all living organisms, and including bacteria. And they basically use these molecules as a communication platform. And each bacterial species has evolved its own chemicals that they secrete and have receptors for, and they organize themselves into communities based on these responses, and they can organize their activities based on the chemicals that are present. So you can think of this as languages, and each species has its own unique language or chemical signal that we're monitoring. They're only produced when the cells are alive, and you can tell who's present based on this. And the molecules are very similar to pheromones that higher level organisms are producing. So these are small molecules for the most part that we're tracking and measuring. What we've been able to do is study a bacterial strain called Pseudomonas that's very common in pathogenic and infections. We've collected isolates from hundreds of patients that have had this infection and grown the cells, and calculated out from our experiments that each cell is secreting thousands of these QSMs each hour into that local environment as part of their communication. And so the concentration of these molecules is actually very high in a sample. So if you collect a sample from a patient that's showing symptoms, you can measure these molecules if you have a receptor that binds to those particular targets. We've also tested the response of the bacteria to antibiotics. So you know that you can track the treatment, because once you have the infection, we've been able to introduce antibiotics and see that the level goes back down over time as the cells die. So those are the applications we're looking at specifically for using these QSMs as a new set of biomarkers for infection diagnostics this way. The other key to this technology is being able to detect these QSMs directly in a biological sample without any sample preparation. And so this was an important part of the IP puzzle for QSM diagnostics, is that there's an electrochemical potential window in which you can measure biological samples such as urine, sputum, blood, ear exudates, and detect these molecules without any interference. And so we were able to test out that we can see these QSMs in this window with zero sample preparation. So directly placing these samples onto a sensor surface and doing the measurement. And that's very important for point of care and low-cost diagnostics, is to have such a simple platform. So this is a picture of our instrument that we currently have in a number of veterinary clinics around the US and starting to place in Europe as well. It's very simple, small handheld. You don't have a size scale indicating here, but it's the size of a television remote or a Roku remote or a glucose meter. And you plug in a disposable single-use cartridge. You can see where the sample's placed. That instrument plugs in via USB to a computer, and you get your test results in 2 minutes on your screen. That tells you the yes, no for the particular bacteria that we're detecting, as well as the concentration of the biomarker that you can then track during subsequent visits for treatment. So to date, we've been focusing, as I mentioned, on the Pseudomonas test. And this is particularly important for dog ear infections, as there's about 9 million of those annually, and about 20% of these ear infections are caused by Pseudomonas. And Pseudomonas is the worst actor because it's very virulent, and if the infection is being caused by Pseudomonas, it turns into a chronic or recurring infection very, very easily. So over 50% of recurring and chronic infections were caused by Pseudomonas. They also actually changed the physiology of a dog ear so that eventually the dog needs surgery if the infections aren't treated properly. To date, we've been able to get several dozen samples mailed in, ear swab samples to us at QSM, and been able to identify based on this process, and we validate by sending out part of that sample to a reference laboratory to do the traditional cultures. And as I mentioned, we are now testing in these laboratories using this concept as well. So that's the starting point for the first product. Our plan is by the end of this year to launch our second product, which is for urinary tract infections. And there, we'd like to broaden the number of causes, bacterial causes that you can detect. And so there's 7 bacterial species that commonly cause urinary tract infections. And these 7 represent over 98% of UTI infections in cats and dogs. And we'll be creating a cartridge that can have 7 sensors on it that detects each of these molecules. So the way the system works, and this ties in very well with Aptamer Group, is we have this array of electrodes on our cartridge. We have the QSMs that are secreted out, and on each electrode in our array, we have an aptamer or optomer that's immobilized. And so when the target molecule interacts with the optomer, it binds, and the way we check for this binding is using electrochemistry. So one end of the optomer is attached to a gold electrode surface so that it's functionalized, and you can imagine it as a tether, this moving around DNA. On the far end of the optomer sequence is an electroactive molecule or a redox label. And it's like a tether ball moving around in solution, bound to the surface. Each time it interacts with the surface, it can transfer an electron, and we measure that current that gets produced. When the target molecule binds to the optomer, the interaction frequency of the redox label changes, and that in turn changes the current that we measure. So we can have, say in this example, 4 electrodes, each one having a unique optomer attached to it, and each one measured independently and see what the concentration of those targets is, and that then tells you what's causing the infection. So now I'd like to talk a little bit about what we've been doing with one of the specific optomers that was provided by Aptamer Group to us. This one is to detect an antibiotic called moxifloxacin, and it's valuable for human applications in that if you do have a serious infection, provide this antibiotic as an IV drug. It gets metabolized by the body very quickly. So you want to make sure that the dosing in the blood is at a high enough dosage that it will kill the bacteria, but not too high where it could cause damage to the patient. So what we did was we attached this functionalized optomer onto our electrode. You can see the 4 electrode slides, the cartridge that we have. So we have the same moxifloxacin optomer on each one of those. These were hand spotted as an initial test, so they're immobilized. And the way the experiment ran is we dip the sensor into a sequential series of solutions. So we'll dip it in, leave it in for a few minutes, move it on, put it into the next solution, and so on and so forth. And what we were able to detect is that we can detect to very low concentrations in phosphate buffer solution as a starting point. We're very happy to be able to get down to 10 micromolar concentrations this way. You don't need any external referencing, but for this case, because we've hand spotted, we start with a blank sample as the first one, and we can normalize the results, and then we can see how the current signal changes as you're going from sample to sample that has a different concentration of the moxifloxacin in it. And what we did was side by side a number of these cartridges, and you can see a very clear difference if you go from blank to blank to blank versus blank to sample to sample to sample. And so this was the first set of data that we we're able to demonstrate. We also have some preliminary results directly in human serum samples. So we took human serum that we purchased, ran it through a 3 kD filter to remove any cells and proteins that are left behind, put in the moxifloxacin, and this is the results you see on this slide are a 1-to-1 dilution of that material in PBS, as well as directly unfiltered serum. You can see that we can detect down to 100 micromolar in the unfiltered and down to 50 micromolar in the 1-to-1 filtered serum samples. So this is very promising as a first experiment. What we're doing now is further optimizing this. So this is just a simple starting point. We also looked at the reaction time. So how quick is the response of these immobilized optomers on the surface to the sample? And you can see that it's immediate. So even as soon as we put the sample down and take a measurement, that molecule has already interacted, and you can distinguish the difference between the various concentrations in the samples. And you might have some fluctuations over time, but the difference between them is always the same. And so as we go to an inkjet printing approach and have a known definite quantity of aptamer each time on each electrode, this will be even more reproducible. The other thing we're starting to look at is how much aptamer do you immobilize on the surface. So to further improve the limit of detection and the dynamic range, what we can do is attach different quantities of the aptamer or optomer onto the electrode surface. And you can see here that the largest response comes from an 800 nanomolar sample being placed that then reacts and binds the aptamer to the surface during the preparation of the sensor. So this is going to continue to go forward and further showcase this. We're excited to be working with the Aptamer group to showcase all of these results. We look forward to getting the optomers for our other small molecule QSM targets to build out our UTI products and other products in our pipeline as we go forward. So just a little bit about where that path goes. I already mentioned the UTI product that's in development. Currently, we're also looking at parasitic tests, so small things like hookworm, roundworm, giardia, various fungi and yeast. They also produce QSMs and virulence factors that could be tracked this way. The nice part is on the companion animal side, there's no regulatory constraints the way there are on the human side. But once we prove out this new testing platform, we do plan to move into livestock and agriculture applications, as well as human point of care testing in other markets. We're also happy to partner with companies that are looking for handheld solution like this for their particular application needs. In terms of where we're at currently, right now this instrument is being sold, the MSRP is about $500 US to the veterinary clinics, and then the cartridges are priced very competitively. So these are much cheaper than sending out for a culture. The Pseudomonas test starts at $40, and we expect the UTI test will be $70 a cartridge to the veterinary clinic. And then the veterinarian can upcharge and sell to the pet owner at the price that they think is appropriate. So thank you for your time. That's what I wanted to tell you guys about today around QSM Diagnostics.
Thank you, Dr. Bunker and Dr. Gerlach, for that interesting presentation. I hope that everyone listening enjoyed the webinar and found it informative. We'll now be moving on to the last part of today's webinar, the question and answer session. Just as a reminder, please feel free to continue asking questions using the tab at the left of your screen. Simply hover over the speech bubble icon and click to ask your question. So to kick things off, the first question I have is, are there any advantages to using optomers over other technologies such as antibodies, protein scaffolds for assay development. Perhaps, David, if you could start us off with this one.
Yeah, no problem. So there's a number of advantages. One of them I mentioned in the slides at the start of the presentation is around being able to tailor the aptamer selection approach to generate aptamers that are specific to a particular target through the counter selection and selection approach. But there are other advantages when it comes to downstream production. So, for example, oligonucleotide synthesis is a well-trodden path, and it's quite readily scaled now so that you can actually produce tens to hundreds of grams of aptamer and optomer in a matter of weeks. So scaling up assays for the likes of veterinary diagnostics or for things like COVID detection is a lot simpler with aptamers and optomers than it is with any other platform. You've also got the advantage that you can site-specifically label them, so with fluorophores or reporter groups or indeed functional groups to attach to biosensors, as we've demonstrated here.
Thank you for answering that one. The next question we have is, are there circumstances where optomers might not be a suitable tool for use in an assay? Perhaps, Edgar, this might be suitable for you.
Sure. So I'd say it really depends on the type of assay you're trying to run. Having worked with several different diagnostic platforms over the years, I can tell you that there's scenarios where optomers are the best or even the only option sometimes. So, for example, for our QSM platform, antibodies cannot be used. The way the signal gets transduced when there's a binding event requires a small flexible molecule. So on our platform is designed really to use an aptamer and optomer as the recognition elements. And so that's how we made that particular decision. Maybe David can chime in on using optomers for other types of assays.
Yeah, sure. So I mean, you're quite right. And one of the other things that's particularly challenging is depending on the matrix, for example. So, you know, aptamers are really only going to work very well in aqueous environments. So if you have a non-aqueous solvent, for example, it's very unlikely that an aptamer will work in that. Also, environments that are highly rich in nucleases can be problematic, but there are ways around that issue by introducing modifications into the aptamer. So a lot of the problems around biosensors we can address in one way or another by adjusting the chemistry.
Great, thank you. Next question I have for you is, you mentioned small molecules, proteins and cells as targets. Have you found any target types that aren't compatible with Optimer selection systems?
I guess I'll take that one. It's a good question. So there are ways of generating aptamers to almost any sort of target, but targets that we find particularly difficult are things with lots of repeating monomer units. So, for example, lipids or carbohydrates, for example, where you have a lot of flexibility but not a lot of structural conservation. They can be quite difficult molecules for aptamers to get hold of and specifically recognize. Now, again, there are ways around that by introducing functional groups, so it's not by any means a hard and fast rule, but generally speaking, they are more difficult than, for example, proteins or small molecules.
Thank you. Edgar, I believe this next question is targeted to you, asking, can QSM's diagnostic platform be applied to other diseases such as cancer or COVID-19?
Yes, definitely. We saw the one example here in the webinar where we started to test for antibiotics. It really comes down to having identify the right biomarker. And as long as there's a validated biomarker that is important for a specific disease, We can work with Aptamer Group to develop the aptamer or the optomer that goes onto our sensor and integrate that into a product. So I'd say if the folks listening, if you have an idea or an interest in applying our platform to a different disease, reach out to us. We're happy to have that conversation and see if there's a partnership that we can develop. We've just chosen to focus on the animal health market and bacterial infections as our initial platform. We can definitely work with you to look at other areas.
Thank you. The next question I have is, how would you minimize off-target effect of aptamers during the selects process?
Again, excuse me, that's a good question. It's actually quite a complicated process, but at the same time fairly straightforward. So we actually minimize off-target effects by speaking to our customers and basically trying to detail those off-target effects as far as possible, and then take them into account through either counter selection, as I mentioned in the early slides. So that's removing aptamers that bind to a protein that might be of concern for cross-reactivity, or introducing a complex matrix, for example, plasma. So in doing so, you generate aptamers that are specific for your target, but don't cross-react with any of those other proteins that might be present in plasma. You can also go a little bit further than this if you have specific targeting requirements in animals, for example, by using an in vivo selection approach, but that's seldom used. We don't use that unless absolutely necessary. Counter selection is usually sufficient.
Thank you. I have another question that might be relevant for you, David, and that is it possible to fuse aptamers to monoclonal antibodies, for example, nanobodies?
Yeah, that's a good question. The simple answer is yes. So there's a lot of work been carried out around the conjugation of other molecules to antibodies, for example, in antibody drug conjugate development. And you can essentially use the same processes to attach an aptamer to an antibody. So you could, for example, have a cysteine side chain in your antibody and put a compatible functional group on the end of the aptamer and use it to conjugate to the antibody in that way. So simple answer is yes, you can.
Great, thank you. Next question is, despite having so much opportunities with aptamers, could you explain perhaps why there's limited products available in the market compared to antibodies, and what might be the reasons for this, and perhaps how we can overcome these hurdles?
Yeah, so I'll address this question first. Having worked in the diagnostic space for over 15 years, I've thought about this question myself quite a bit. And I found two reasons really for where we're at this time. One of the main things, having talked to a number of other companies in this space as well, is that the large established companies are somewhat resistant to change. And if they've developed a pipeline of antibody development and expertise with working with antibodies, it's an uphill battle to get them to change to a different product, even if it is better. The other scenario that you would have is a new company or a startup that you would think would be able to utilize aptamers on their diagnostic platform. But there's a number of challenges there. So first you have to find a useful biomarker or target molecule and validate it. And after that, then you have to successfully market the product. And I have come across at least a few companies that have some amazing technologies that work really well, but end up failing in the market. And because the biomarker wasn't adopted by the consumer, by the end user. And so we've seen Successful aptamer diagnostics unfortunately fail for those reasons. I think by focusing on what the target is and what the problem is, and then working backward and using an aptamer we hope to be successful and be one of those big companies that we can point to with doing that. But I'm starting to see other companies now in this space as well. Now that there are new biomarkers that don't have antibodies and new diagnostic testing platforms that are getting developed that might work better with aptamers than with antibodies.
Great, thank you. Another question's come in asking, will an aptamer bind to a non-denatured protein?
Simple answer again is yes. We actually prefer non-denatured proteins. They tend to be a little better behaved in the selection process. That's not to say that we can't work with denatured proteins. So we have had projects where we've looked at aptamers as reagents for use in Western blots, for example, so the protein would be denatured there. But the vast majority of projects that we undertake are on non-denatured, native, and even complex proteins. So again, simple answer is yes.
Great, thank you, David. Next question is there any information on how optomers perform in terms of sensitivity and specificity for small molecules and protein quantification when compared to LC-MS?
So we haven't done any direct comparisons with LC-MS. Obviously, mass spec is a very sensitive technique, so the likelihood of an aptamer-based assay ever being as sensitive as something like mass spec is slim. But the other side of that coin is that mass spec, for example, is a very expensive process that requires very large labs, very large equipment, so it's by no means simple to use or portable, which is where technologies like this really come into their own. So, we're not going after the super sensitive end of the market. We're after the rapid testing, rapid detection, and rapid quantification.
Great, thank you. Edgar, I believe this next one's for you. How sensitive is your Pseudomonas test for BAL as compared to PCR-based assays?
That's a very good question, and one I can get into a lot of technical detail on, but I'll try to give you the short answer in that it's not as sensitive as PCR, and we never intended to be as sensitive as PCR, where you can find single molecules or a needle in a haystack. Our detection is really geared toward physiologically relevant levels. So, and I'm assuming when you say BAL in the question, it's referring to bronchial lavage samples, where they're not sterile samples. You're collecting fluid from the lungs and analyzing it for signs of infection. So in that scenario, that's not a sterile sample. You have a lot of bacteria naturally present in your lungs. And what I can tell you from what we've tested is we can detect physiologically relevant concentrations just as well as PCR does, and much faster and more accurately than standard culturing techniques, because we're detecting the QSMs associated with these bacterial targets. And we get similar specificity to what you would achieve with PCR, but just a very different application space in terms of that. But we can do this, say, with a 2-minute test. I think that might tie into another question I saw. We can get down to, say, 10 to the 4th CFU per ml directly from a sample, and you get your answer in 2 minutes with that level of detection.
Great, thank you. I believe I have a follow-up question asking if your test is strain specific And is it only available for a bacterial infection? And how about parasites or viruses?
Mm-hmm. So we specifically chose to start with bacteria. That's where I first got involved with QSMs. Fungi and parasites also make QSMs, and we've already identified those targets associated with several fungal and parasitic infectious sources. We have 2 sensors in our development for fungal targets already. We have sensors in our product pipeline that we plan to make for parasites as well. Bacteria don't produce QSMs, so while we can make a sensor for detecting viruses, they can't be detecting QSMs. They have to detect either the virus itself or something else related to the virus. But yeah, so that's what we can do with this approach.
Thank you. Another question, again, possibly for you, is the sensor reusable?
Generally, it's not reusable. So what happens is that there's a few different versions, but the sensor that I presented in the webinar is geared toward a single use, where the sample remains on the sensor cartridge, and then you would throw it out and grab a new one. We are working in our R&D team on a continuous monitoring product where the sensor could be used, say, for a week. That's going to require some different and more complex surface chemistry, but for the moment, these are really designed as a single-use sensor tests.
Thank you. I've probably got time for a few more questions. So the next one I have is, can Optimers be used as a point-of-care test for toxic trace metal analysis?
Yeah, I would imagine so. The approach that we've taken here, looking at aptamers and optomers that undergo a conformational change when they bind to a target, theoretically that can be applied to anything. Obviously, there is going to be a limit on that because the sensors like this require a conformational change, and very, very small molecules like metal ions are probably not going to induce a significant conformational change. So the sensitivity might be limited, but in theory it is possible.
Thank you. And perhaps the final question, has there been any work done with saliva?
That's a very good question and very topical. The answer is yes. So we've actually been working on our COVID aptamer-based test using saliva and nasal swabs as a matrix. And obviously you have to deal with the viscosity issues and so on, but the aptamers do recognize their target in that matrix, yes.
Great, thank you. Well, I'm afraid that is all we have time for today. Thank you, Dr. Bunker and Dr. Golak, for that interesting presentation. And I really hope that everyone listening enjoyed the webinar and found it informative. If you have any other questions from today's session, please feel free to email myself at editor@selectscience.net and I'll follow up with your questions. Remember, you can also download related resources in the tab to the left of your screen, which includes a certificate of attendance. And if you'd like to listen again to today's webinar, or perhaps invite a friend to listen, it'll be available to watch on demand in just a few days' time. Goodbye, and thank you once again for joining us.